Good morning, everyone. So a couple of reminders. As Mike has written here on the board, no discussion on Monday. You can come to a Tuesday discussion section if your normal day is Monday. And also, please refer to BSpace for information about the exam. There was an announcement posted yesterday. I'm still getting a few emails from people with questions from the class. Please attend office hours. If you can't make it to my office hours, please attend your GSI's hours or those of Mike Meehan, okay? All right, so today we're going to talk about anaerobic respiration and fermentation. And this is the last part of Chapter 9. So this is basically rounding out our discussion of how cells make energy, or not make it, but are able to harvest energy from food that comes in and break it down through oxidation and reduction reactions, generating ATP and ultimately ATP, but also some important cofactors in the process. All right, so the idea, so you recall from earlier in the week that we've been talking about aerobic respiration, where cells are able to metabolize sugars in the presence of oxygen to produce ATP. But it turns out that, and this is true for most cellular respiration, that this occurs in the presence of oxygen. But it turns out that certain organisms and some cell types can carry out metabolism in the absence of oxygen. And we want to talk a little bit about that today. So if you recall, glycolysis, which we talked about on Monday, is a process that can produce ATP with or without oxygen being present. Oxygen is not required, and that's because in glycolysis, we're starting with a 6-carbon sugar, right? We're starting with glucose, and we're breaking it down to a 3-carbon molecule called pyruvate, and that is a process that doesn't lead to full oxidation of the glucose. Full oxidation would result in the production of carbon dioxide and water, right? And that only happens in oxidative phosphorylation. So glucose is a process that can occur under aerobic or anaerobic conditions. Now, if there's no oxygen around, it turns out that, as we said, some organisms and cell types can couple glycolysis to a further cycle, either fermentation or anaerobic respiration, to produce ATP. So these would be organisms that aren't using oxidative phosphorylation. All right, so anaerobic respiration is a process that uses an electron transport chain, so very similar to what we've been talking about with aerobic respiration. But in this case, we have an electron acceptor, an ultimate electron acceptor that is not oxygen, not O2. And there's some really interesting examples of this in biology. And for anybody that's interested, there's some nice discussions of this on the internet. And there's been a lot of interest in these processes for a long time because of the use of microbes to break down waste products that humans are producing, and also more recently to use microbes to produce other kinds of compounds that might be useful as, for example, biofuels. So this is an area that I think there's been a real resurgence in biology of interest in these kinds of pathways with microbes. So for example, this kind of process, anaerobic respiration, would take place in organisms that live in an environment that lacks oxygen. And so these guys have an electron transport chain, but they use something like sulfate as the ultimate electron acceptor. And in that case, those are organisms that end up producing hydrogen sulfide. Think about it. Hydrogen sulfide is the final product. So very interesting examples of this in biology. Okay, and then fermentation is a process that uses phosphorylation, and this would be substrate level phosphorylation, instead of an electron transport chain to generate ATP. And again, in both cases, these would be coupled to glycolysis. So in basically all of the examples of respiration that we're talking about, Glycolysis is on the front end and does the initial steps of breakdown of glucose. And then pyruvate can feed into these other pathways depending on the type of organism or cell that we're talking about, and also whether or not oxygen is present. Okay, so let's talk a little bit more about fermentation. So, as we said, so this consists of glycolysis plus reactions that serve to regenerate NAD+, which can then be reused by glycolysis. So this is a cycle that can occur again and again. And you might remember that in glycolysis, we're producing 2 molecules of NADH, right? We're also producing 2 molecules of ATP. That's the net yield in glycolysis. And so if we didn't have a way to, if these organisms that don't carry out aerobic respiration had no way of regenerating NAD+, then once the NAD+ supply was depleted, they would basically stop respiring. And so fermentation takes on that job of regenerating the NAD+ that can then feed back into the glycolytic pathway. And there are 2 common types of fermentation: alcohol fermentation, which yields ethanol, and lactic acid fermentation that yields lactic acid. And you should just be aware that, again, there's lots of different types of fermentation. So biology is very diverse in this regard. So there are microbes that can carry out fermentation leading to different kinds of compounds. And again, this is an area where there's a lot of current interest. And I would say that, we really don't understand, we don't have a full understanding of the microbial world by far. And so there's a lot of pathways out there that are probably still yet to be discovered. I just recently saw a paper in the literature where somebody had figured out a natural microbe that makes gasoline. Imagine, gasoline, right? And so I think that this is a really interesting area going forward of discovery in biology. So let's talk a little bit about alcohol fermentation. So this is a process in which pyruvate, so remember this is that 3-carbon compound coming out of the glycolytic pathway. Pyruvate is converted to ethanol, which is a 2-carbon molecule, in 2 steps with the release of a carbon dioxide molecule. So one of the carbons comes off as carbon dioxide. Now, alcohol fermentation is interesting historically because humans have known about this pathway and have been able to use it for literally thousands of years. And so you know for sure that alcohol fermentation by yeast is used in brewing, in winemaking, and in baking, right? And it's also used for things like, let's just stick with those examples. So brewing, winemaking, and baking. And this is a process that takes advantage of a natural ability of yeast cells to ferment sugars into alcohol, and in the process releasing carbon dioxide. So the next slide shows this pathway and gives you sort of an overview of how it's coupled to glycolysis. So again, glycolysis, we're starting off with a 6-carbon sugar. We're going through this pathway 10 steps, right? We're generating 2 molecules of ATP by substrate-level phosphorylation, and we're ending up with 2 molecules of pyruvate for every molecule of glucose that enters the pathway. 2 molecules of pyruvate. And these pyruvate molecules, if they go through the alcohol fermentation pathway, the pyruvate is then converted to ethanol in 2 steps. In the first step, we split off carbon dioxide, 2 molecules of carbon dioxide, because we have 2 molecules of pyruvate that are input here, and then we end up with 2 molecules of acetaldehyde. And acetaldehyde is then converted enzymatically. Of course, these are enzyme-driven steps, right? They're enzymes catalyzing these steps, converted to ethanol, which is shown over here. And this is a oxidation reduction, a redox reaction. And in this reaction, 2 molecules of NADH are oxidized to 2 molecules of NAD+. So as this in this reaction, is this the reducing agent or the oxidizing agent? I heard both, but oxidizing is right? Because it's leading to oxidation of NADH to NAD+, but it's becoming reduced itself in the process, right? This is a reduction of acetaldehyde to ethanol. Okay, let's see if I was going to tell you anything else here. Okay, so again, remember that this is these ATPs are being produced by substrate level phosphorylation. The NAD+ that's regenerated is then able to re-enter the glycolytic pathway. So this is really important because it keeps this cycle going. So notice that all of the pathways that we're talking about in respiration, whether we're talking about aerobic respiration or fermentation in this case, and it's true also with anaerobic respiration, these are all cyclical pathways, right? They form products that can then regenerate compounds that can re-enter the pathway and keep it going. Although, of course, there has to be an initial input for animals of a sugar source like glucose. All right, and then just a note here. So the carbon dioxide that's coming off here. So this is basically a waste product, right? It's a byproduct of this alcohol fermentation pathway, but it's actually very critical for baking, for example. It provides the bubbles that lead to yeasted breads being able to rise, right? It's by the production of this waste product, CO2. Okay, and then that's alcohol fermentation. There's another kind of fermentation that I want to mention, and this is called lactic acid fermentation. And in this type of reaction, we have pyruvate, again, being reduced by NADH, not to NADH. That's a little mistake in the notes. By NADH, forming lactate as an end product. And there's no release of carbon dioxide in this case. All right, and so this is a sort of a one-step reaction, and it's used by certain fungi and bacteria that are employed by humans to make food like cheese and yogurt. And it turns out that, very interestingly, human muscle cells can also use lactic acid fermentation to generate ATP when oxygen is scarce. And that would be a situation that would occur when under strenuous exercise, when you first start exercising, the cells initially will be burning glucose and generating ATP through that pathway faster than they can acquire oxygen to take it through oxidative phosphorylation. And so under that type of situation, then the pyruvate enters this lactic acid pathway, producing lactic acid. And so if you feel muscle burn in your legs, that's often due to the production of lactic acid. Although I read your book says, I didn't realize this, but we were taught that when you get sore muscles from after strenuous exercise, it's often due to the buildup of lactic acid. It turns out that more recent research has suggested that the lactic acid is actually beneficial to muscles, and what is causing the muscle pain is due to a buildup of potassium ions in the muscle. Okay, right. So let's look at that pathway. And so again, here's the glycolytic pathway yielding pyruvate, and look how simple this is. It's really just one step, right? Pyruvate being converted to lactate over here, still a 3-carbon compound, so there's no production of CO2 in this pathway. But again, we get the regeneration of NAD+ in the process. So this is, again, very important for keeping glycolysis going. It provides the NAD+ that can then enter the glycolytic pathway and result in ATP production. All right, so it's worth thinking a little bit about the efficiencies of these pathways. So again, this is a redox reaction, right? So is pyruvate the reducing agent or the oxidizing reagent in this reaction? Oxidizing, right? Okay, exactly. So again, because what's happening is that NADH is being oxidized to NAD+, and in the process, pyruvate is being reduced to lactate, right? So another example of oxidation and reduction. And then it turns out that when muscle cells carry out this pathway, lactate is then recycled in the liver. It's recycled back to pyruvate in the liver through another cyclical metabolic pathway called the Cori cycle. And we won't go into that in detail, but you should know that this isn't just thrown away. It's actually recycled again in the body. And so this is really the simplest type of fermentation, because it's really just one step from the end product of glycolysis to the end product of this fermentation pathway. So just one enzymatic step right here, regenerating NAD+. And so the other thing to note is that overall, this is a much less efficient way to make ATP than oxidative phosphorylation, right? Because we're not fully oxidizing the products of glycolysis. We're just getting to lactate. And so ultimately, we're only making these 2 molecules of ATP overall for each turn of this cycle. How does that compare to oxidative phosphorylation? Do you remember how many ATPs are made? I hear some murmuring, but I don't hear the number. But it's about 38, right? Your book says about 38 molecules of ATP that are made per molecule of glucose that we start with, that goes through all the steps, right? Goes through glycolysis, then goes through the citric acid cycle, and finally through oxidative phosphorylation, generating ATP, right? So we end up with about 38. So this is a much less efficient pathway. And so not too surprisingly, what we find is that most of the time, this is a pathway that's carried out by very simple organisms like bacteria or yeast that don't have enormous energy requirements, right? If humans were to try to function using lactic acid fermentation, we would quickly run out of energy, right? Because our cells just need too much energy. All right, so let's compare fermentation and aerobic respiration a little bit. This is what we sort of summarizing some of what we've talked about. So in both cases, these are processes that start with glycolysis to oxidize glucose, and as we'll see in a few minutes, also other organic fuels, and they oxidize them to pyruvate. So we end up with pyruvate, and these are processes that have different final electron acceptors, right? So in the case of fermentation, we have an organic molecule like pyruvate or acetaldehyde is involved in as the final electron acceptor. Certainly in aerobic respiration, oxygen would be the final electron acceptor, and of course, not listed here, but as we said, in anaerobic respiration, we would go all the way. We would still have an electron transport chain, but we would have some other molecule as the final electron acceptor, like sulfate. All right, and then this is important to know that cellular respiration is a lot more efficient at producing ATP. So we get up to 38 ATP molecules per glucose versus only about 2 ATP molecules per glucose in fermentation. All right, and so different cells have different abilities to use these pathways. We can define obligate anaerobes as organisms that carry out fermentation or anaerobic respiration only, and they actually can't survive in the presence of oxygen. So, these are organisms that have basically evolved to grow in an environment where they have no access to oxygen, and if they get exposed to oxygen, they get poisoned and they're killed, right? So they can't survive in the presence of oxygen. More commonly, yeast and many bacteria are what are called facultative anaerobes. So these are bugs that can survive using either fermentation or cellular respiration. So, yeast are a great example of this, right? They can either ferment sugars, or they can grow aerobically. They can grow in the presence of oxygen. So they're not obligate anaerobes. And our muscle cells, it turns out, are also facultative anaerobes, right? Human muscle cells, because they can grow aerobically. Obviously, they can grow in the presence of oxygen. But if oxygen is limiting, they can carry out lactic acid fermentation to generate ATP. And in a facultative anaerobe, pyruvate is a fork in the road. It's a fork that leads to two alternative breakdown pathways, catabolic pathways. Can go either direction. Can either go into the citric acid cycle, or it can go into a fermentation pathway. Oh yeah, I wanted to mention too that certain cells are, for example, in the brain, carry out only aerobic oxidation by pyruvate and not fermentation. So it can sort of go either way. So these are bugs that can only ferment, and then there are cells like in our brain that can only carry out aerobic respiration. So it can go both ways. So this is a slide. This is another figure from the book that sort of illustrates this idea that glycolysis is going on in the cytosol, right? It's a cytosolic pathway generating pyruvate, and this pyruvate can then enter either of these pathways, at least in a facultative anaerobe, right? It can either undergo, if oxygen is around, can enter this aerobic respiration pathway by entering the mitochondrion if it's a eukaryote, and making acetyl-CoA and going into the citric acid cycle we talked about on Wednesday. If there's no oxygen around, then this molecule will undergo fermentation. And we talked about two examples where this ends up as either ethanol with the production of a CO2 or as lactate. Right, so under these conditions, anaerobic conditions, pyruvate is diverted from the citric acid cycle, and it itself serves as an electron acceptor to recycle NAD+, right? That's what we showed. So this can basically, doesn't show it in this picture, but remember that this is then regenerating NAD+, that can then go back and recycle into the glycolytic pathway here, right? So this is a cycle. So it's very interesting to think about the evolutionary significance of the glycolytic pathway, because as you can see from our discussion so far, glycolysis is a pathway that occurs in basically all organisms, right? And so the thinking is that it probably evolved in ancient prokaryotic cells before there was oxygen that was present in the atmosphere of the Earth. And so, people have looked, and sort of there's a fossil record for bacteria and microbes in addition to a fossil record for other kinds of organisms. And going back in that microbial fossil record, we can go back about 3.5 million years to find what are currently the oldest known types of microbes that we can see. And it's thought from various other kinds of geological studies that at that time in the Earth's atmosphere there wasn't very much oxygen around. So the guess is that those would be organisms that were mostly growing anaerobically. They didn't have access to much oxygen, so they used glycolysis and then various fermentation pathways to generate the energy they needed to carry out cellular processes. And then at some point there was the evolution of cyanobacteria, which are bacteria that are photosynthetic and they can actually generate oxygen. And it was really the production of oxygen by those early microbes that led to the buildup of oxygen in the atmosphere, and then much later the evolution of aerobic respiratory pathways. So glycolysis and the citric acid cycle turn out to be major points of intersection with other both catabolic and anabolic pathways in the cell. And so we've been discussing glucose as the input into glycolysis. Oops, sorry, little typo there. As the fuel for cellular respiration. But as you probably know, free glucose is not usually what humans and other animals are eating directly in their diet, right? They're not eating free glucose. Instead, most calories are coming in through fats, proteins, sucrose and other disaccharides, and also from various kinds of polysaccharides that are consumed, right? Those are really the starting materials. And so these can all be used by cellular respiration to make ATP in the kinds of, in these pathways, because they can be used to first generate glucose that then enters the pathway. But importantly, they can also be used to make all the molecules that are necessary in the cell. So only some of the food that we eat is actually burned as fuel. Other food that we eat is broken down and then used as the starting material to make things like DNA, RNA, proteins, and the fats that go into cell membranes. So these catabolic pathways have to be very versatile. So we've sort of drawn them focusing on the generation of energy, but you should be aware that at every step in these pathways, molecules can be coming in or out, right? So, and we'll see we'll sort of see how that works. The idea is that these breakdown pathways funnel electrons from many different kinds of organic molecules into cellular respiration. Glycolysis can accept a wide range of starting carbohydrates. Again, glucose is what enters the pathway, but there are steps upstream that can lead to breakdown of more complex molecules and sugars to glucose so that they can then be enter the glycolytic pathway. In the case of proteins, these have to be digested first to the individual amino acids. And these amino acids, fix that, can feed glycolysis or the citric acid cycle. So, for example, starch is hydrolyzed in the digestive tract down to its, so you remember when we talked about starch, right, being long chains of sugars, so there are enzymes in the digestive tract that digest those polysaccharides into individual sugars, and if necessary, isomerize the sugar or change it to glucose if possible so that it can enter the glycolytic pathway. And then glycogen is a long polysaccharide that occurs in the liver, stored in the liver and muscle cells, and is hydrolyzed to glucose to supply energy between meals. Some amino acids that are produced here are also not burned in these pathways, but they're actually recycled to make other proteins, right? So this is, again, a point where there's a divergence of input of these molecules. They can either be used in anabolic synthesis of other proteins or other molecules, or they can be used in catabolic pathways to make energy. And cells are constantly having to figure out which pathways need to be more active, right? If the cell needs more energy, then there's going to be more respiration going on. If the cell needs to produce certain proteins, then these amino acids will be siphoned off for that purpose. And the amino groups that are removed by deamination are waste products, right? And so they're excreted in the form of either ammonia or urea. And then fats are digested to, so remember the structure of fats, right? Remember we have that 3-carbon glycerol molecule with fatty acid tails that are esterified to the hydroxyls of glycerol. And so when fats are digested, they're digested down to glycerol, which can then be converted to glutaraldehyde 3-phosphate. Remember, that was one of the intermediates in the glycolytic pathway. So in that case, we enter the glycolytic pathway a little bit downstream of where glucose would enter. for fats. And then the fatty acids can actually be broken down by a process called beta oxidation. And beta oxidation breaks them down into 2-carbon molecules that are used as they become the acetyl groups for acetyl-CoA. So they can actually enter the citric acid cycle directly. They don't have to go first through glycolysis. They go directly into the TCA cycle through acetyl-CoA. And an oxidized gram of fat produces more than twice as much ATP as an oxidized gram of carbohydrate. So that's why when we eat a lot of fat, it has a lot more energy. It's a lot more energy intense than eating carbohydrate. Why is that? Does anybody know? Think about the chemical structure of fats and carbohydrates, the fatty acid tails. Lots of hydrogens, exactly, right? So there's lots. They're sort of higher up, effectively, on that. If you think about that energy diagram, right? So there's more oxidation that can go on, and so there's more energy to be produced from those fatty acid tails than we get from sugars that are farther down they're more reduced overall than fatty acid molecules are. Okay, so this is a diagram from your textbook that just illustrates the fact that we have fats, carbohydrates, and proteins that are all entering the glycolytic pathway in some cases, or, as we said, in some cases entering directly into the citric acid cycle through acetyl-CoA or sometimes through other intermediates in the citric acid cycle. So this is a really important point because I don't want you to, we've been really focused on glucose as the starting sugar, but I want you to know that and to remember that the real starting materials are actually these much more complex molecules, and they're being broken down by many different kinds of enzymes so that they can be used as the starting materials in these pathways, glycolysis, the citric acid cycle, and then ultimately in oxidative phosphorylation. And so this just sort of illustrates the fact that glycerol enters as glyceraldehyde 3-phosphate right here. The fatty acids are broken down into 2-carbon molecules that acetylate CoA and enter the citric acid cycle. Sugars can enter at various steps in glycolysis. They can either, if they can be converted easily to glucose, they enter at the top, but if not, they can sometimes enter farther downstream in the glycolytic cycle. And then proteins have several possibilities. They can be converted to pyruvate and enter the pathway at this step, acetyl-CoA, or other intermediates in the citric acid cycle. And again, in this case, ammonia is coming off as a waste product. All right, so as we said, it's very important to be able to break down complex molecules. It's also very important for cells to be able to build up complex molecules. And the body uses small molecules to make these much more complex molecules like nucleic acids, proteins, and lipids through other kinds of metabolic pathways that often start with compounds that can go either way. They can either be burned as fuel or they can be used as building blocks. And these are coming most of the time from food or from glycolysis or from the citric acid cycle. So very important central, remember that complex figure I showed you several lectures ago that showed huge mess of metabolic pathways in the cell, but really in many ways this pathways we've been discussing this week are right at the heart of that, right? Because providing a means for cells not only to harvest energy, but also to make the starting materials for all the other molecules that need to be synthesized. So for example, in humans, our bodies have to make roughly half of the 20 amino acids that are necessary to make proteins, right? We make those, in our bodies. And the rest of the amino acids are called essential amino acids. We can't make them biosynthetically. We have to consume them in our diet, right? So those are the essential amino acids. And if you remember the dihydroxyacetone phosphate that we talked about, one of the intermediates in the glycolytic pathway, and there's an isomerase enzyme that converts it to glyceraldehyde 3-phosphate, right? And we talked about the fact that DHAP, the dihydroxyacetone phosphate, doesn't directly enter the glycolytic pathway, but it's not just simply there. It's not just sort of a useless molecule in the pathway, because it actually can be converted to one of the major precursors of fats. And so when energy is very abundant, for example, and there's lots of glycolysis going on and energy is being produced abundantly, some of the dihydroxyacetone phosphate in the glycolytic pathway can be siphoned off and used to synthesize fats for storage of energy. So as you can imagine, there's lots of feedback going on in these pathways to regulate whether cells are primarily going through cellular respiration or whether they're siphoning off molecules for biosynthetic pathways. And the most common mechanism for this kind of control is feedback inhibition. So remember, we talked about feedback inhibition when we discussed enzymes, and the idea is that if you have a pathway where we have a whole series of enzymes catalyzing multiple steps in the pathway, ending up with a final product, if the final product becomes very abundant in feedback inhibition, it goes back and binds often to the very first enzyme in the pathway, and either often not completely inactivates it, but certainly downregulates its activity. So there's a slowdown of the pathway, and that allows cells to then use accumulated product molecules, and then when those get depleted, they no longer inhibit the first enzyme in the pathway, and there's a ramping up again. So it's a very nice way of easily controlling the flux through a pathway depending on how much of the product of the pathway is present. It's basically supply and demand. And so in the broadest sense, if ATP concentration is low, then cellular respiration speeds up to make more of it, and when there's a lot of ATP around, then cellular respiration slows down. Control of these catabolic pathways is based primarily on regulation of these enzymes, and often this is done, as in feedback inhibition, at points in the pathway that are strategic. In other words, they're at key points where a pathway sort of decision has to be made about whether a molecule is committed to a particular pathway. Okay, so here's a great, this is kind of a nice example of this that just shows that a key point of regulation in the glycolytic pathway is at the step that's catalyzed by the enzyme called phosphofructokinase. You remember we talked about this enzyme previously. We abbreviated it PFK, phosphofructokinase. This is an enzyme that is important in basically catalyzes the third step in the glycolytic pathway. And it's important because this is the step at which the cycle becomes committed. So at this point, the fructose bisphosphate that's produced is then committed to entering the cycle. And so not too surprisingly, this enzyme is very highly regulated. So for example, if there is a lot of adenosine monophosphate around in the cell, AMP, that's indicative of an absence or a reduction in the amount of ATP around. That tends to stimulate this enzyme. So if there's not a lot of ATP around, there's a lot of AMP around, the hydrolytic product that's produced after ATP is hydrolyzed in the cell. This binds to the enzyme and stimulates its activity. We need to make more ATP. We're going to ramp up glycolysis. And in contrast, if there's a lot of ATP around that's coming out of these various pathways, this will go back and bind to phosphofructokinase and inhibit its activity, right? So these have converse effects. And likewise, citric acid that's coming out of the citric acid cycle, if the citric acid cycle is really cranking and we're making a lot of citric acid, the citrate will come back and also bind to phosphofructokinase and inhibit its activity. So there's multiple ways that this whole pathway can be regulated by an enzyme that's really near the very top of the process, right? And so that's a very classic example of feedback inhibition. And we know a lot about how this works in the case of phosphofructokinase because we know there are many available molecular structures of this enzyme that have been solved using X-ray crystallography, and we know where these inhibitors and stimulators bind on the enzyme and how they tweak it structurally so that it's either in a more active or less active form. All right, so the rest of the slides that are in the handout are basically just kind of review of what we've said earlier and underscore, I think, some of the key points that we've talked about this week. So, for example, this is just showing you that when we start with glucose in the glycolytic pathway, we end up, for every molecule of glucose, we end up with 2 molecules of pyruvate, and we get 2 molecules of ATP and 2 molecules of NADH. And remember that this is the overall output, but you recall that in glycolysis we consume 2 molecules of ATP early on to phosphorylate the sugar as it's entering the pathway and make it more competent for hydrolysis. And then we generate 4 molecules of ATP later in the pathway. So the overall production is 2 molecules of ATP and 2 molecules of NADH. And then in the citric acid cycle, we have the input is 2 molecules of acetyl-CoA. And by the way, again, this is for every molecule of glucose that we start with. Entering the citric acid cycle, going through the cycle, and yielding 2 molecules of ATP and 6 of NADH and 2 of FADH2. And these are what go on into the oxidative phosphorylation pathway and lead to the production of ATP by oxidative phosphorylation and not by substrate phosphorylation like in these cycles here. Okay, and then finally, this is just something that reminds you of what we've seen before, where we have in the oxidative phosphorylation pathway, which would be going on over here in the inner mitochondrial membrane, we're pumping protons out into this intermembrane space, and they are then able to re-enter the mitochondrial matrix by flowing down their concentration gradient through ATP synthase. And this is a very cool little motor, like we talked about last time, that's basically spinning around physically and catalyzing the direct synthesis of ATP from ADP and inorganic phosphate to produce ATP over here. Okay, so be sure that you review all of these pathways, study your notes, do the example problems in the back of the book, because I think they're very helpful for making sure that you are understanding the concepts of metabolism. And if you have questions, come to office hours, and we'll try to make sure we answer your questions before the exam. Have a nice long weekend. I'll see you on Wednesday.